Chapter 5, Microscopic Observation of Cells, by Brenda and me. As we know, plants are eukaryotic, multicellular, and have a biome depending on certain characteristics. Plants contain chloroplast, which is essential to the process of photosynthesis. Photosynthesis is what allows plants to acquire energy from the sun. Plants also have cell walls. The specimen we have examined is Elodea. Here you can see our close-up picture of the Elodea sample at 400x magnification. So in this next picture, the parts of the cell are labeled. Those parts are the chloroplast, cell wall, nucleus, and central vacuole of several different Elodea cells. The chloroplasts are easily distinguished as the green parts of the cell. The cell wall borders the outside of the cell, and the central vacuole, which helps control water intake, is shown as the clear, or at least not green, part of the cell. And the nucleus is the brownish color. You can tell from on the right, that brown part anyway. So, animal cells. They're eukaryotic, multicellular, and they have different environmental niches. Some examples could be the tundra, forest, desert, savanna. There are lots of other ones too. So a distinguishing characteristic is that they are heterotrophic. So this pretty much means that they don't make their own food. They get energy from consuming something else. So when it says they rely on other organisms for sustenance, they get their nutrients from those organisms. They don't just photosynthesize or something like that. The specimen that we observed were cheek cells. Here you can see our close-up picture of the animal sample at 400x magnification, my cheek cells. You can notice that the clumping of cells is a little hard to view, but that might be because we applied a bit too much methylene blue dye. Still, the important structures can be viewed. In this next picture, I have labeled the parts of the cell. Those parts are the cytoplasm, plasma membrane, and the nucleus. Fungi. Fungi have eukaryotic cells and can be either unicellular or multicellular depending on the specimen. An example of a unicellular fungi is yeast, which we will be showing you in this presentation, and a multicellular fungi could be mold or mushrooms. As mentioned in the introduction, fungi live in damp, moisture-rich areas, and they have cell walls. So we have our specimen yeast now. The clarity of the yeast is really not so great at 40x. Still can't see very much at 100x, but once you get up to 400x, you can really see everything. So it gets greater as the magnification increases from the 40, 100 to 400. And at the last magnification, you can see the shape of the cells is somewhat rounded. Bacteria. So bacteria are prokaryotic, unicellular, and they have the environmental niche of soil, water, and the human body. The distinguishing characteristics are aerobic, as Brenda said earlier. That means it needs oxygen for its cellular respiration. It has a cell wall, and most are not harmful, but some of them are pathogenic, so they can cause diseases. The specimen we have is Anabaena. The archaea is actually also important here. It used to be just known as bacteria, but then they broke it into groups, bacteria and archaea. Archaea is kind of the same thing, but it's not as abundant as bacteria, and it's found in extreme environments. So first, we have displayed the bacteria that were previously prepared for our viewing in lab. So you can see that we have Spirillum, Bacillus, Coccus, and Staphylococcus, which are pretty much supposed to be named after their shapes, like Spirillum is spiral, and Staphylococcus is obviously a kind of coccus. So these bacteria have distinguished shapes, and these were all viewed at 1000x magnification. And so that's it for that. And then we have the Anabaena, which was observed at 400x. Protists. Again, protists are eukaryotic, and they could be either unicellular or multicellular depending on the specimen. They live in almost any environment that contains liquid water, and I'll just skip past the points that Brenda made in the introduction. And also under the distinguishing characteristics, you could note that there was chloroplast and photosynthesis listed. Chloroplast is essential to the process of photosynthesis, which we went over earlier. Protists use it to acquire energy on their own, and they are not heterotrophic. So, like we said earlier, that means that they make their own food. They don't depend on other organisms for that nutrients. So I'm going to skip past this specimen thing real quick. Okay, here we have a little picture. It's kind of irrelevant, but I just wanted to bring in the point that there's primary producers, because a lot of protists are actually primary producers in the ecosystem. So, for example, plankton in the ocean is a primary producer. So in other words, they can be the organisms that the heterotrophic organisms depend on for their sustenance. But not all protists aid other organisms in maintaining homeostasis, because certain protists actually cause the very serious diseases like malaria or sleeping sickness. Anyway, the specimens we're observing are trypanosoma in the rat blood smear and algae. So first we have the algae, which we observed at 400x magnification. And then we have trypanosoma, which is here at 400x magnification. So it's important to wear gloves while you're handling the blood smear. Just mentioning this because you don't want to cause infection of any kind, since this is one of those protists that could cause a serious disease. So that's pretty much it for our sum up of the different kingdoms and some of the factors about each of the things that we observed. So I hope you enjoyed, and that's it. We'll see you next time.